皆様、大変お待たせいたしました。ただいまより2020-2021年若手外国人農林水産研究者表彰ジャパンアワードを開会いたします。私は本日司会進行を務めます若手真由子と申します。どうぞよろしくお願いいたします。それでは開会にあたりまして農林水産省農林水産技術会議会長小林義雄よりご挨拶申し上げます。小林会長、よろしくお願いいたします。
農林水産技術会議会長の小林でございます。主催者として一言ご挨拶いたします。本日は第15回若手外国人農林水産研究者表彰の表彰式をご覧いただきまして誠にありがとうございます。例年は会場に直接お越しいただいていましたが、新型コロナウイルスの感染拡大の影響により昨年は表彰式が中止となり、また今年も厳しい状況が続く中、初めての試みとしてこのように動画配信の形で表彰式を開催することとなりました。ご協力いただきました皆様に御礼申し上げます。本表彰式においては政府の総合科学技術イノベーション会議の上山隆弘議員や独立行政法人国際協力機構の佐藤正上級審議役よりご祝辞をいただいております。また本表彰の選考委員会の岩本座長をはじめ委員の方々には受賞候補者選出の審査において熱心なご議論をいただきました。この場を借りて改めて厚く御礼申し上げます。さて、近年飢餓貧困や栄養不良、気候変動、越境性病害虫などの地球規模の課題がますます深刻なものとなっています。また新型コロナウイルスの感染拡大はこれらの問題にも暗い影を落としています。このような中、本年9月に国連食糧システムサミットが開催され、また12月には東京栄養サミットの開催が予定されるなど、世界は持続的な食糧システムの在り方について模索を続けているところです。農林水産省としましても、食料・農林水産業の生産力向上と持続性と両立をイノベーションで実現する緑の食料システム戦略を本年5月に策定しました。これをアジアモンスーン地域の持続的な食料システムのモデルとして打ち出すなど、各国との対話も行いながら取り組みを進めているところです。このような中、農林水産分野における国際研究協力の推進を加速し、またそのための国際ネットワークの形成や土台となる開発途上地域の研究者の能力構築を図ることが従来にも増して重要になっています。この若手外国人農林水産研究者表彰は、茂田茂元農林水産技術会議会長の特使により2007年に開始され、今年で15回目を迎えました。本表彰は開発途上地域の農林水産業及び関連産業に関する研究開発について、その一層の発展及びこれに従事する研究者の意欲向上に資するよう、すでに優れた功績を挙げている、または将来の技術革新につながる優れた研究業績を上げつつある若手外国人研究者を表彰するものです。国際的にもジャパンアワードとして高い認知を得ており、食料安全保障や気候変動といった地球規模の課題に立ち向かっていく若手外国人農林水産研究者にとって大いに励みになるものとされています。過去の受賞者におかれましては、本表彰の受賞契機として自国における評価や認知度がさらに高まり、またその後の論文執筆や研究成果の普及などの精力的な取り組みを通じて、ますます活躍の場を世界へと広げていっているものと承知しています。このような先輩研究者に続き、本年に表彰いたしますのは、 各分野で優秀な実績を上げられ、将来を大きく期待される3名の方々です。インドからマラヤ大学上級講師のサハデブ・シャルマ氏、エジプトからベンハー大学助講師のハラガマル・アリアリ・エル・ダウス氏、ガーナからクアメ・エンクルバ科学技術大学講師のジェームス・セウトラ・カバ氏、以上の3名の受賞者の方々に心からお祝いを申し上げます。受賞者の方々におかれましては、本日の受賞を契機としてそれぞれの専攻についてさらに磨きをかけて、開発途上地域における農林水産業および関連産業の発展に貢献していただければ幸いです。同時に我が国の研究者との交流を一層深めていただき、我が国と各地域の架け橋としてさまざまな問題の解決に向けて取り組みを推進されることを期待しています。来年には新型コロナウイルスの感染が収まり、我が国で受賞者の方々とお会いできることを心より記念し、主催者からのご挨拶とさせていただきます。受賞者の皆様、改めておめでとうございました。
小林会長よりご挨拶申し上げました。続きまして、内閣府上山隆弘様からご祝辞を頂戴いたします。上山様、よろしくお願いいたします。
Good afternoon. I am Takahiro Ueyama, Executive Member of the Council for Science, Technology and Innovation, CSTI, at Cabinet Office of the Japanese government, which serves as the headquarters for creating the comprehensive and basic government policies for science and technology under the direct leadership of our Prime Minister and our Minister for Science and Technology. For today's award, I'm very pleased to learn that the recipients are 3 excellent researchers from India, Egypt, and Ghana. On behalf of the CSTI, I heartily congratulate you on your success. I'd like to take just a moment to tell you a little more about CSTI and why we wish to support researchers like our award recipients today. The Council is responsible for creating Japan's 5-year national plan for science and technology. In the 5th basic plan, we propose a new concept called Society 5.0. Society 5.0 represents an advanced human-centered society in which the integration of cyberspace and physical space is to be realized through such state-of-the-art technologies as artificial intelligence, the Internet of Things, robotics, and big data, in all of which Japan has significant strength. In the 6th basic plan, which has launched this April, described concrete policies in relation to Society 5.0 under the following 3 pillars to realize this concept. Number 1, transforming our country into a highly sustainable and resilient society through the fusion of cyberspace and physical space. Number 2, advancing knowledge creation, which designs a new society and becomes the source of value creation. And number 3, accelerating innovative human resources to support a new society. Agriculture, forestry, and fishery is one of the fields to which the Society 5.0 will make the most important contribution. Our new technological advancement will help agricultural operators who worry about labor shortages. We are working on automating agricultural industry and achieving labor savings by utilizing robot combine harvesters and other technologies. However, in the field of agriculture, as in so many other fields, because globalization is progressing, Japan alone cannot solve the serious social problem we now face. It is definitely necessary for us to share our issues, foster human resources, and utilize brain across international borders. For this reason, it is very important for excellent, motivated researchers to cooperate no matter what their country of origin is. We strongly expect that with this prize, these excellent young researchers who are awarded today will not only work for their own country, but also become a bridge between each of their countries and Japan, and enhance the research collaboration among Japanese scholars and their counterparts throughout the world. We hope that today's award create energy and momentum to fuel the future partnership for many years to come. On behalf of CSTI, I sincerely hope that you will have many more successful years in your own field, and we become new leaders promoting in collaboration with Japanese researchers world research activities that will improve the world we live in, the people of your countries, the people of our own, far into the future. Congratulations for today.
上山様、ありがとうございました。続きまして、独立行政法人国際協力機構上級審議役佐藤忠様からご祝辞を頂戴いたします。佐藤様、よろしくお願いいたします。
独立行政法人国際協力機構 JICA の上級審議役の佐藤忠です。JICA を代表して受賞者の皆様に心よりお祝い申し上げます。受賞者の皆様は私たちが生きていく上で欠かせない農林水産業やそれを取り巻く環境の保全について素晴らしい研究成果を上げておられます。国際協力を通じて地球規模の課題の解決に取り組んでいる JICA として皆様のご尽力と貢献に大いに敬意を表します。さて、この機会に JICA の活動についてご紹介するとともに JICA が皆様のような学術研究者の方々との連携を重視していることを申し上げたいと思います。JICA は日本の政府開発援助 ODA の実施機関です。約150カ国に拠点を置き、各国の政府や関係機関の方々と共に働き、信頼で世界をつなぐというビジョンのもと、技術協力、有償資金協力、無償資金協力といった援助手法により、開発途上国の発展のために尽力しているところです。ここでは人間の安全保障の一層の推進、質の高い成長の促進、さまざまな主体との連携の強化など、日本政府が定めた開発協力大綱などに基づく具体的な事業を展開しています。JICA の実施する事業の中に、地球規模課題に対応する科学技術協力があります。具体的には、地球規模課題対応国際科学技術協力プログラム SATREPS というものです。これは、環境エネルギー、生物資源、防災及び感染症などをはじめとする地球規模課題に、日本と開発途上国の大学研究機関が研究開発を通じて取り組むものです。このSATREPSを通じて、国際科学技術協力の強化、地球規模課題の解決につながる新たな知見や技術の獲得とイノベーションの創出、そしてキャパシティディベロップメントへの貢献が期待されます。今回の受賞者のお一人が、かつてこのSATREPSプロジェクトの一つに参加され、その際の成果も今回の受賞に寄与していると伺い、JICA としても大変嬉しく、また誇らしく思います。さらに JICA は最新の学術研究の成果を現場の事業に活用できるよう、GILCAS や海外の大学など国際的な研究機関と密接なコミュニケーションや情報交換を行っています。時には JICA 事業の専門家としてこれら機関の研究者からアドバイスをいただき、それぞれの国の開発事業を行うこともあります。このように皆様は JICA の活動において欠かせないパートナーです。私たちのさまざまな活動において皆様のような若く有望な研究者の方々と連携して開発途上国のさまざまな課題に取り組んでいくことを期待しています。皆様が研究においてさらなる成功を収められることを記念し、私からのご挨拶とさせていただきます。
佐藤様、ありがとうございました。続きまして、選考委員会座長岩本睦夫より若手外国人農林水産研究者表彰の審査経緯をご報告申し上げます。岩本座長、よろしくお願いいたします。
選考委員会を代表して本年の若手外国人農林水産研究表彰ジャパンアワードの選考経過についてご報告をいたします。農林水産省におきましては2007年に農林水産技術会議会長が優れた若手外国人研究者を表彰する制度を創設いたしました。この制度は開発途上地域の農林水産業および関連産業に関する研究開発に貢献した研究者の一層の意欲向上を図るため、問題の解決につながった研究や将来の技術革新等につながる研究において優れた研究業績を上げた若手外国人研究者を表彰するものです。この賞の受賞者には農林水産技術会議会長賞に加え、モタイ・ジルカス賞として副賞の奨励金が授与されるとともに、日本に招聘され表彰式で受賞講演を発表する栄誉が与えられることになっております。近年国際社会では急激に増加している世界人口を支えるための食料システムの構築と気候変動への対応など、地球規模課題の解決に向けた研究がますます重要性を増しております。さらに新型コロナウイルスの感染拡大により、食料農林水産業における課題も一層顕在化し、ポストコロナ社会への対応の検討も求められているところでございます。農林水産技術会議の在職中に本省を創設され、以来現在も強力にご支援をいただいているモタイ茂様元農林水産技術会議会長の先見の明とご英断に対し、選考を担当した者として深甚なる敬意を表する次第でございます。さて、開会のご挨拶にもございましたとおり、新型コロナウイルスの世界的な感染拡大の影響により、2020年は表彰式を開催できませんでした。本年も受賞者の方々を招聘できておりませんが、本年はこのような動画配信を通じて表彰式が開催されますところ、2020年の受賞者の方々3名と2021年の受賞者の方々3名について審査の経過をご紹介いたします。まず第一次審査でございますが、2020年は岩永勝国立研究開発法人国際農林水産業研究センター理事長、永井俊彦農林水産技術会議事務局研究総務官、夏秋恵子東京農業大学副学長、原田信彦株式会社読売新聞東北統括本部次長、丸山清明国立研究開発法人農業食品産業技術総合研究機構元理事、村上秀則一般財団法人食品産業センター理事長に、座長である私岩本を加えた7名の審査委員により書面による審査を行いました。本年2021年は岩永氏に代わり小山治国立研究開発法人国際農林水産業研究センター理事長、また永井氏に代わり山口純一郎農林水産技術会議事務局研究総務官が審査委員となりました。またその他数名の委員におかれましては所属の変更等がございましたが、これら計7名で書面による審査を行いました。第2次審査は2020年は8月26日に農林水産省会議室において、また今年2021年は8月26日にオンラインにてそれぞれ行いました。第2次審査に先立ち本委員会としての考え方を整理し、受賞者が同一の国から昨年に続き連続して出ることは可能な限り避ける。第一次審査の獲得点数で ボーダーラインを決めて、その上にあるすべての候補者を広く第二次審査の議論の対象とする。同一分野の候補者が複数いる場合には1名に絞り、可能な限り多くの分野の候補者を選出する。同一の受賞国や組織からの選出は可能な限り避ける。また受賞者は審査員全員の合意に基づいて決定する。以上の視点で各候補者の選考を行いました。研究分野は多岐にわたり、レベルの高い研究や有用性の高い研究が多く、議論は白熱しましたが、厳正な審査の結果、次の方々を審査員全一致で受賞者に決定いたしました。まずは2020年に27名の応募者から選ばれた受賞者3名とその研究成果への公表をご紹介いたします。まず1人目はインドからインド農業研究委員会中央塩利土壌研究所のサラスワティプラ・ラクシュトマイヤー・クリシュナム・ルティ博士の従来法および分子育種法を使った多園性稲品種の開発であります。クリシュナム・ルティ博士は従来法および分子育種法を使った稲の多園性を向上させる先駆的な研究を行い、育種選抜された多園性の稲品種に幅広い遺伝的変異を作り出しました。また稲のさまざまなマッピング個体群を使用し、生殖段階の多園性に関する新規 QTL 遺伝子の特定に大きく貢献いたしました。本研究は最新の育種法を用いて品種登録までを進め、商品化に至るまで尽力している点や、気候変動下においてインド以外の地域への波及効果が高い点が評価されました。2人目はタイからチュラポーン研究所のクワンラシシリカンジャナ博士の効率的な農業流域管理に向けた新しい微生物ツールによる畜産由来汚染源の識別法であります。シリカンジャ博士は従来型 PCR などの DNA ベースの検知方法を用いて糞便由来の微生物源追跡マーカーを開発し、バクテリオファージ DNA の分析を行うことにより流域水域の汚染源の識別を可能とする迅速分析法を確立し、タイの農業流域における畜産由来汚染の実態を明らかにしました。新たに開発した方法は農業のみならず環境への取り組みに応用できる汎用性の高いツールであり、タイのみならず水質汚染対策に取り組む国々でも広く利用できる点が評価されたところでございます。さらに3人目はジンバブエからジンバブエ大学のエディモ・ガスラ博士のジンバブエの農村地域における高品質タンパク質トウモロコシの強靭性強化と高栄養食品や飼料としての利用であります。ガスラ博士は雑草であるストライガに抵抗性があり、高収量高タンパク質一大雑種のトウモロコシの品種開発に農家とともに取り組みました。またトウモロコシ生産だけではなく、栄養価の高い食品や家畜の飼料として活用するビジネスに取り組み、実用的な成果を上げました。本研究は雑草害に苦しむ生産現場の課題に対応し、地域経済への貢献度も高い研究である点が評価されたところであります。次に本年2021年に35名の応募者から選ばれた受賞者3名とその研究成果への公表をご紹介いたします。まず1人目はインドからマラヤ大学のサハテブ・シャルマ博士のアジア太平洋地域における気候変動を緩和する自然に基づく効果的解決策のためのマングローブ林のブルーカーボンの研究であります。シャルマ博士はリモートセンシング等を用いて気候変動が沿岸堆積物のダイナミクスやブルーカーボンサイクルに与える影響に関する研究に取り組み、 マングローブ林の環境保全機能の定量的評価法を開発しました。気候変動による海面上昇等の課題に対してブルーカーボンの重要性を示しており、研究成果の普及性の高さや国際的な研究協力の実績が評価されました。2人目はエジプトから原ガマルアリエル・ダウス博士のダイレクトフィルター PCR を用いた牛伝染性リンパ腫ウイルス感染の新規診断検査手法の確立であります。ダウス博士が開発したダイレクトフィルター PCR 方式を使用した新しい検査方法は、血液の付着したフィルター紙を PCR 用のテンプレートとして用いることにより、従来の手法より精度が高く、安価迅速簡便な診断を行うことが可能になりました。この検査手法は設備や人材が不足している開発途上国では特に有用と考えられ、今後の実用化と普及が期待されるとともに、発展途上国への貢献度の高い研究である点が評価されたところでございます。3人目はガーナからジェームス・セウトラ・カバ博士のグルイシンディアとの観察システムにおけるカカオの窒素栄養でございます。カバ博士は熱帯土壌でのカカオ栽培のアグロフォレストリーシステムにおける窒素循環に関する研究に取り組み、マメ科樹木グリリシンディアとの最適な観察方法を示しました。また本研究は従来推定が難しい窒素循環をデータで示し、小規模カカオ農家の観察の実践に貢献いたしました。提案された観察方法は、ガーナをはじめとする熱帯地域の小規模カカオ農家の経営及び生計の改善に役立つことが期待され、普及性の高い研究であること点が評価されたところでございます。以上、受賞した研究課題は先端的な研究手法、あるいは普及実用化に対する地道な努力を重ねられた結果であり、開発途上地域の農林水産業に貢献する優れた成果であります。それぞれの国、地域のみならず、地球規模で解決が急務となっている問題に正面から取り組み、成果を上げたものであることをご報告いたします。選考委員会の全委員を代表して、受賞者の皆様には心からのお祝いを申し上げたいと思います。このジャパンアワードは若手研修者の奨励の賞でありますので、受賞者の皆様はここでとどまることなく、将来に向かってさらなるご発展をされることを強く希望しております。受賞者の皆様、そしてご家族の皆様、本当におめでとうございました。
岩本座長よりご報告申し上げました。受賞者の方々には表彰状および$5000の報奨金、モタイジルカス賞を授与するとともに、表彰式出席のため日本に招聘しています。昨年と今年は新型コロナウイルス感染症の影響で受賞者の方々の招聘は行っておりませんが、この度の栄えあるご受賞を心よりお祝い申し上げます。ここで受賞された皆様の記念撮影の時間とさせていただきます。受賞者の皆様、誠におめでとうございました。それではここで休憩を入れさせていただきます。 それではお時間となりましたので、受賞者の皆様よりご講演を頂戴したいと存じます。はじめに従来法および分子育種法を使った耐塩性稲品種の開発と題しまして、サラスワティプラ・ラクシュマイヤ・クリシナムルティ様にご講演いただきます。クリシナムルティ様、よろしくお願いいたします。
First of all, I would like to express my sincere thanks to Ministry of Agriculture, Forestry and Fishery, Japan, and Agriculture, Forestry, Fishery Research Council, Japan, for selecting me for this award. I will be delivering the lecture on research achievement on development of salt-tolerant rice varieties through conventional and molecular breeding approaches. At a global, approximately 830 million hectares of land is affected by salt, whereas in India, 6.73 million hectares is affected by salt. Out of 6.73 million hectares of land, 2.96 million hectares is under saline stress, 3.77 million hectares is under sodic stress. Approximately the 22% of yield loss is due to salinity stress, and approximately 40% of rice is occupied in salt-affected areas of 6.73 million hectares. It is also estimated that annual economic loss of $1,666 million is due to saline stress, and $2,000 million is due to sodic stress. Rice is sensitive at two stages. One is at early seedling stage, another is at reproductive stage. So many QTLs has been reported for early seedling stage, and we need to explore other novel QTL for early seedling stage to pyramiding these QTL to enhance the salinity tolerance. Whereas in case of reproductive stage salinity tolerance, very less number of QTL has been identified. We need to identify the novel and robust QTL for reproductive stage salinity tolerance. Keeping all these research gaps in mind, we collected 9,000 of rice germplasm from National Bureau of Plant Genetic Resources, New Delhi, and we evaluated them for 30 qualitative and quantitative trials. And we developed 2,500 core collection and evaluated them in salt stress for reproductive stage. We identified some lines for reproductive stage 2 and are using for enhancing the salinity tolerance in rice. In one of the projects, we have collected 7,500 rice lines and we evaluated for seedling stage at salinity stress EC of 10 decimol per meter. So we could able to identify 241 rice germplasm as a tolerant. We are using these 241 rice line for our breeding program to enhance the salinity tolerance. Similarly, we have developed a mapping population using different donors. So we have developed the biparental mapping population, and also we have developed the rice panel for identify the QTLs for reproductive stage salinity tolerance. We evaluated them for moderately salinity at EC of 8 decimal per meter, high salinity stress EC of 10 decimal per meter, moderate sodicity with the pH of 9.5 and high sodic stress with the pH of 9.9. In one of the projects, we have identified or collected 500 rice germplasm across the country and evaluated them in seedling stage salinity tolerance, reproductive stage salinity at EC of 10 decimol per meter, and reproductive stage sodicity with a pH of 9.5 and sodicity with a pH of 9.5. Interestingly, we have identified two lines, KLR 270 and 271, are tolerant to all the stages and all the stresses. Now we are using these two lines to decipher the physiological and molecular mechanism for seedling as well as reproductive stage salinity tolerance. As you all we know that labor and water is the main constraint in rice production system. Hence, we are trying to develop the rice varieties for direct seeded rice situation. We developed many number of rice for PSR, and we evaluated them for in farmers field to identify the best one. Interestingly, CSR 80 has given the average grain yield of 7.2 ton in irrigated and puddle condition, whereas in case of DSR situation, CSR 88 was given 6.2 ton per hectare of grain yield. We also trying to identify the novel and robust QTL for salinity as well as sodicity tolerance. Here we used a magic population received from IRRI, and we used the GPS data for market rate association. For seedling stage salinity tolerance, we identify the QTL on chromosome number 1. It is the SKC gene is already reported QTL. And we evaluated this line for sodicity stress with a pH of 9.7. And we also identify the marker trait association on chromosome number 6 and chromosome number 11. This chromosome number 6 is overlapping with the 3 genes. And interestingly, aluminum activated malate gene transport is mainly responsible for sodicity tolerance at seedling stage. And chromosome number 11, finger-like protein as gene is overlapping in a reported QTL. We also used GWAS panel to identify the marker trait association physiological trait. In our studies, the chromosome number 9 has reported the many marker traits association. Totally, we identify the 28 marker trait association for reproductive stage salinity tolerance for physiological trait. In other experiment, we also identify the marker trait association for sodium and potassium homeostasis on chromosome number 1. We also used the biparental mapping population to identify the reproductive stage salinity tolerance, reproductive stage sodicity tolerance. We evaluated CSR 11 into MI48 for 3 seasons under normal situation, the pH of 7.5 and sodic of pH of 9.5. We used 50K SNP genotyping data to identify the QTLs. A major QTL SSI 6.1 on chromosome number 6 with a phenotypic variation of 32%. Once you identify the QTL, we need to introgress the QTL in yielding varieties. So we introgress the Saltal QTL in 2 backgrounds, Sajja 52 and Pisa 44. So we developed the 10 nils of Pusa 44 background and 8 nils of Sajju 52 background. Similarly, the spikelate fertility QTL has been identified in chromosome number 8. We are introgressing this QTL in 3 mega varieties, Pusa 44, PR114 and Sajju 52 using Marcus-Sted backcross breeding. So we are also using stringent phenotyping while selecting the nils. Once you identify the lines or Marcus-Sted lines nils, we need to evaluate it in salt stress location in the farmer's field. We evaluated these lines in 18 different locations. It includes 9 coastal saline and 9 alkaline environment in India. In one of the experiments, we identified the line CSR 46 is adapted to sodic soils of India. We also released this line as a variety. So we also developed the salt tolerant rice varieties through conventional breeding program. First variety CSR 46. It is a medium duration with a long bold grain type. It has a potential of 6.5 ton grain yield under normal stress and 4 ton per hectare of grain yield under salt stress. This is recommended sodic soils of Uttar Pradesh. Second variety, this CSR 56, it is also medium duration with long slender grain type. It has a potential of 7 ton per hectare of grain yield under non-stress, 4.3 ton per hectare of grain yield under salt affected situation. It is recommended to sodic soils of Uttar Pradesh and Haryana. The another variety is CSR 66. It is a medium duration with long slender grain type. It has a yield potential of 7 ton per hectare under non-stress, 4.5 ton under stress situation. It is recommended to sodic soils of Uttar Pradesh and Pondicherry. Another variety is CSR 49. It is also medium duration. It is tolerant up to pH of 9.6 with a potential of 6.5 ton per hectare under non-stress and 4 ton per hectare under salt situation. It is recommended to sodic soils of Uttar Pradesh. Another variety is CSR 52. It is also medium duration but medium slender type. It has a potential of 6.5 ton per hectare under non-stress situation, 4 ton per hectare under salt stress situation. It is recommended to sodic soils of Uttar Pradesh. Another recent variety is CSR 76. It is a long slender with medium duration type. It has a potential of 7 ton per hectare under non-stress, 4.5 ton under salt affected situation. It is also recommended for sodic soils of Uttar Pradesh. Apart from these varieties, we have also developed a genetic stock, CSR 47, CSR 53, CSR 51, CSR 59, Kala Jawha. These genetic stocks are being used by scientific community in India to enhance the salinity tolerance in their interested rice background. These rice varieties covered 1.6 million hectares of salt affected land annually in India. An estimated value produced $4,732 million US. These salt tolerant rice varieties has been adopted by the Land Reclamation Corporation like UPLDC, HLRDC, benefiting 430,000 farmers. With the introduction of these salt tolerant rice variety can save the 0.6 million tons of gypsum annually, and it cost of $250,000 US. It is the impact of salt tolerant varieties for 12 years of period. Finally, I express my sincere thanks to our DG ICR, Dr. T. Mahapatra, Deputy Director General NRM, Dr. S.K. Chaudhary, Director CSSRI, Dr. P.C. Sharma, different funding agency, and to my rice team at CSSRI, Karnal. Thank you very much for their support and encouragement. Thank you very much.
クリシナムルティ様、ありがとうございました。続きまして、効率的な農業流域管理に向けた新しい微生物ツールによる畜産由来汚染源の識別法と題しまして、クアンラウィー・シリカンジャナ様にご講演いただきます。シリカンジャナ様、よろしくお願いいたします。
Hello, everyone. My name is Kwanrawi Sirikanjana. I am a research scientist at the Laboratory of Biotechnology, Jelapon Research Institute, Thailand. I am truly honored to receive the 2020 Japan International Award for Young Agricultural Researchers. Today, I'm delighted to share with you my research work on the topic of novel microbial tools to distinguish fecal pollution sources from livestock for effective agricultural watershed management. The outline of my presentation will be divided into 4 subsections. First, I will introduce you to the situation of livestock and community wastewater pollution in Thailand. In 2020, water quality of rivers and lakes in Thailand is shown in this map, with the orange color as poor water quality and the green color as the good water quality. Fecal contamination, as determined by total coliform and fecal coliform bacteria, is one of the main sources of the water degradation. Poor water quality and fair water quality is a persisting problem in the same water bodies for more than 10 years. And in 2020, Thailand raises about 12 million pigs and almost 7 million cattle, which contribute to wastewater contamination into the environment. The sources of fecal contamination that lead to poor water quality can be divided into two main types: point sources and the non-point sources. The point sources are the sources that can be easily pointed at or identified. These could be animal farm disposal, combined sewer overflow, and wastewater treatment plant disposal. And for the non-point sources, these are the sources that can be more difficult to identify. For example, the sewer pipeline leakage, septic tank leakage, and these are from the underground. And also the rainfall runoff from different types of the land use, such as pasture for animal grazing, domestic pets, wildlife in national parks, urban space. Also, the seabirds and waterfowls are sources of animal fecal contamination, while boats and ships and swimmers are sources of human fecal contamination also into the water. Because some fecal contamination sources are difficult to identify, we need a source-specific marker to trace the contamination sources. Unfortunately, the bacterial fecal indicators, as used in water quality standards in many countries, can indicate only the general fecal contamination, but they cannot differentiate the sources of fecal pollution, whether from human or animals, or each specific type of animals. And because the ability to identify wastewater sources will facilitate the restoration and management of poor water quality, we need another research technique. And that is the concept of microbial source tracking, or MST. Microbial source tracking, or MST, is a research area that can track the fecal water pollution to its origin by detecting the host-specific gastrointestinal microorganisms excreted with the feces into the polluted environment. MST can serve as a tool for discriminating one or more than one pollution sources, for example, the communities, animal farms, wildlife, birds, and waterfowls. MST can therefore be also be used for assessing human health risks to the waterborne pathogen. One of the laboratory techniques that we use to detect the source-specific MST marker is a molecular method called polymerase chain reaction, or PCR. So it starts by collecting a water sample from a river that has been contaminated by fecal pollution, but we don't know who polluted the water. Next, after collecting the samples, the sample is extracted for the DNA. Then the polymerase chain reaction, or PCR, is used, which is a method for amplifying particular segments of DNA using specific primers here in one of the steps of the PCR. And the conventional PCR provides qualitative results in positive, negative results of the marker presence. For example, if the primer that can detect the cow marker is used, when a sample turns positive, that means the water has been contaminated by the cow wastewater. Moreover, the quantitative PCR also delivers copies of MST genes. So they give the results of the gene copies per milliliter of the water. And this is very useful to prioritize the main sources of the contamination. For example, cow may be the main source, but we can also identify the secondary source or the tertiary source as well. Next, I'm going to show you how the microbial source tracking technique can be used to identify the livestock wastewater in the Thajin River in Thailand. For the project of using microbial tools, which are the MST markers, to distinguish fecal pollution sources from the livestock, we study the Thajin agricultural watershed in the central region of Thailand. In the Thajin watershed, wastewater from pig farms was estimated only from the point sources to contribute 35% of the wastewater, both treated and untreated, to the water resources. However, there's still more from the non-point sources. For example, from the rainfalls, they could contribute more pollution from pig and also from cow farms to the water resources. So in this project, we first did the PCR screening to select the best performing PCR primers. So first, we tested 17 specific primers over 1,600 animal individuals according to the actual ratios of density of each animal in the watershed. And the N here in the picture represent the composite sample of one farm. So these are, N is the number of the farms, but it's actually from over 1,000 individuals. And specific primers detect the DNA of bacterial order Bacteroidalis and the bacteriophage crashphage. The best pig and cow specific fecal markers are the pig-to-bac EP here and the BAC3 assays is the name of the specific primers. And these two showed highest sensitivity and highest specificity. And the sensitivity means the ability to detect the target marker in all target samples. And specificity means the ability to detect the target marker only in the target host and not cross-react with the other non-target host, which we don't want. After we know the best MST markers for pig and cow wastewater, we further characterize their performance. This research also investigated human sewage and general fecal sources, but in this presentation, I'm going to just focus only on the pig and cow markers. For the pig, MST marker, both conventional PCR that show just positive, negative result, and the quantitative PCR provide very high sensitivity and very high specificity, and can detect high marker copies in the pig feces. For the cow MST marker, BAC3, both conventional and quantitative PCR provide very high specificity, but relatively lower sensitivity here, but they still have the high marker copies in the cow feces. Next, we used these MST methods for the freshwater monitoring to track the sources of fecal pollution in the Thajin River. This is the rotated map of the river. So the north direction is towards the right. The universal markers, which show the general fecal contamination, were present in all 12 stations at all 4 sampling times. So indicating persisting and continuing fecal contamination. And for the swine or pig specific markers, they were found in all lower river sample. This is the lower section of the sample. And interestingly, for the cow, they show up only in the wet season, and it corresponds to the way that the cows were raised in the open space. Moreover, we found that the conventional PCR could serve as a low-cost pre-screening step for the quantitative PCR method. In the last section, I would like to summarize the output and the impact created by this research project. We published the new scientific knowledge in 6 international publications from this research project. And then we also translated the scientific knowledge into 2 useful documents. The first document is the Thailand Microbial Source Tracking Guideline, which collected the standard operating procedures, or the SOP, for sampling and laboratory protocols. And this is aimed for the laboratory personnel in academic and governmental sectors. The second document is the policy brief that contained a brief summary of the significance of the microbial source tracking research, advantages, recommendations, and costs. This is developed for policymakers responsible for water quality management. Towards the end of the project, we also held a dissemination workshop to present how microbial source tracking method can help for pollution management, especially the non-point sources, which are difficult to identify and manage. We were successful to have participants from both the academic sector and the governmental sector. And their very useful comments during the workshop were integrated into the guidelines in the policy brief, as I just showed. Our research stories has also been recognized in the news media as well. And so here come my last slide. For this research, we aimed to pinpoint fecal pollution sources from livestock farming, especially pigs and cows, and dense residential communities along the 325-kilometer Tajin River located in central Thailand. And the microbial markers were developed and validated to specifically track contamination from pigs and cows using DNA of the bacterial order Bacteroidalis, and from the communities using DNA of the bacteriophage crayfish. These microbial tools also reveal spatial and temporal patterns of the contamination from the different sources. So before I finish, I would like to acknowledge the funding sources from national and international organizations. Thank you again to the Ministry of Agriculture, Forest and Fisheries of Japan and Japan International Research Center for Agricultural Sciences that recognize my work. And also thank you all for your attention.
シリカンジャナ様、ありがとうございました。続きまして、ジンバブエの農村地域における高品質タンパク質トウモロコシの強靭性強化と高栄養食品や飼料としての利用と題しまして、エディモ・ガスラ様にご講演いただきます。ガスラ様、よろしくお願いいたします。
Greetings to you all. My name is Dr. Edmo Gasura, Head of Department of Crop Science and Associate Professor of Plant Genetics, Biotechnology and Breeding, University of Zimbabwe. I am going to present to you on the work that I did on strengthening quality protein maize resilience and utilization as nutritious food and feed under smallholder farming systems. The consumption rate for maize in Southern Africa is very high. If you look at Zimbabwe, it's almost at par with Zambia, and they are all following Malawi, where the consumption rate per person is very high per year. However, in these countries, specifically Zimbabwe, The demand, which is appearing here in this red line, is higher than the production. And there are several factors that are contributing to that, including the parasitic weeds that are found in this region. We have Striga hemontheca and Striga asiatica. And these parasitic weeds, they are very common in soils of poor soil fertility. And they are widely distributed in Africa. As you can see that the entire of Africa is covered by this parasite. The yield loss is estimated to be at 7 billion United States dollars annually in this region, especially where the infestation rates are very high, as shown on this slide. Our team that I was leading started breeding work specifically focusing on quality protein maize, which is high in lysine and tryptophan, the amino acids which are rare in most diets found in rural communities in Africa. So we evaluated these materials for Striga resistance using different strategies like the agar-gel assay. We also evaluated these materials for Striga resistance in the greenhouse, as you can see on this picture, that some materials they are having Striga asiatica and some they do not have. And when we went into the field, it was very interesting. Some materials like this first one, it didn't have any cob. But some of the materials like this one is tolerant, yet some materials could even produce two cobs, suggesting some improvements in the breeding that was done. We also developed 2 breeding tools for Striga resistance to aid our future breeding programs, and we published 2 papers, one in the International Journal of Genomics, focusing on the genome-wide association studies, and we also published another one in Genetic Resource and Conservation and Utilization, focusing on the grouping of materials to enable heterosis breeding. The quality protein maize that we developed, most of them, they yielded even much better than the Czech. If you look at MQ623, it was yielding as high as some common Czechs. And we had a student who was also focusing specifically on that area. The quality protein maize seed that we produced was also fed to broilers. Very interesting to notice that these broilers, they grew very well and they produced the meat, which was very tasty, high in yield, and it was liked also by some consumers who ate and tasted some of those Bosch veals, the species of those indigenous chickens. And we presented this work in the international conference with that student, Thelma. From the maize straw, we didn't throw it away, but we also tried to use it in the production of mushrooms, and we managed to produce healthy mushrooms from the maize straw. And we worked with a number of farmers. As you can see that I'm standing there in front of a number of farmers, all the activities that we have been doing. So this project, we made several linkages with these different stakeholders. For example, the Capital Feeds, it's a private feed company, Mukushi Seeds, which is now selling some of these varieties. The extension agencies under the government department, development agencies, specifically Ruforam and the World Hunger Ilfe, those are the donor agencies who supported this work. We trained over 500 farmers in field days. 3 international conferences were conducted. 1 international newsletter by the American Society of Agronomy. We also appeared in that newsletter. We went on to influence policy, and we proposed that the Grain Marketing Board should separate the QPM and the non-QPM materials. And this separation, it also enables the addition of a premium price to the quality protein maize. And we proposed what is called VCC, varieties compulsory for companies, to say each company that is existing in the country should sell at least one nutritious crop, and we're focusing on quality product maize here. And there is a paper that we produce in the African Crop Science Journal, focusing on those policy issues. So in conclusion, we managed to produce the QPM varieties, and the QPM varieties, they were fed to broilers, and the broilers, they responded very well. And the maize straw from these QPM varieties was used in the production of mushrooms. A total of 8 publications were produced, 9 students were trained, 2 breeding tools were developed, and upscaling plans, they existed through the policies which were suggested, and also the industrial linkages that were made. So this slide is just showing some of the publications that we produced. In conclusion, I would want to thank a number of donor agencies who supported our work, some partners who also provided us with the germplasm and the training that I received. And I would not want to end this without thanking JICA for the recognition of the young scientists, especially in this region of Africa, specifically Zimbabwe. I'm very grateful to the support that I got from JICA and the recognition. Thank you very much.
桂様、ありがとうございました。続きまして、アジア太平洋地域における気候変動を緩和する自然に基づく効果的解決策のためのマングローブ林のブルーカーボンの研究と題しまして、サハデブ・シャルマ様にご講演いただきます。シャルマ様、よろしくお願いいたします。
Distinguished guests, ladies and gentlemen, firstly, I would like to thank Ministry of Agriculture, Forestry and Fisheries and JIRCAS for presenting me Japan International Award for year 2021. And I would also like to thank for giving me opportunity to share output from my research on mangrove forest, blue carbon, for effective nature-based solutions to mitigate climate change in Asia Pacific region. During this presentation, I will discuss mangrove ecosystem services, nature-based solutions, role in climate change mitigation, deforestation drivers, and its related carbon emissions under the introduction section. And I will share the results from selected studies and take-home messages. As we know, mangrove forests provide several ecosystem services such as provisioning, regulating, and cultural services. And I have listed down some important ecosystem services, such as fishery, habitat for food security, wildlife habitat for biodiversity conservation, and improving the water quality through trapping the fine sediments, and storm protection from cyclones through wave attenuation. During last decade, mangrove carbon storage potential also recognized as an important ecosystem services because it can play an important role in climate change mitigation and adaptation. Now, nature-based solutions are designed to address major societal challenges such as food security, climate change, water security, human health, disaster risk, social development, and economic development. Now, this figure shows the few examples of nature-based solutions, and mangrove forest here listed as one of the nature-based solutions in providing the benefits to the communities. Now, mangrove can also play an important role in achieving sustainable development goals. So according to the UNDESA report, mangrove are very important in achieving SDG 13 climate change, SDG 2 zero hunger, SDG 15 life on land, and SDG 1 no poverty, SDG 8 economic growth, and SDG 5 in gender equality. Now, let's discuss the role of mangroves in climate change mitigation and adaptation. So, the blue carbon tank was first coined in 2009. Carbon sequestrated in seagrass, salt marshes, and mangrove ecosystems called blue carbon. Now, this is an classic paper published in 2011 entitled Mangrove Among the Most Carbon-Rich Forest in the Tropics, which was published in Nature Geoscience. And this paper showed that the mangrove can play an important role in climate change mitigation and adaptation. According to this figure, carbon stock potential varies across different ecosystems. And it is very clear that the mangrove forest has highest ecosystem carbon stocks as compared to other ecosystems, like almost 80 to 85% of the carbon stored in the soil, which is due to anaerobic condition of the soil, which either stops or slows down the organic matter decomposition process. And therefore, carbon can be stored in soil for decades to thousands of years. And therefore, we can say that the mangrove ecosystem sequester and store significant amount of coastal blue carbon from the atmosphere and ocean, and hence are now recognized for their role in mitigating climate change. Now, despite mangrove forests provide several ecosystem services, but in the last 50 years, 30 to 50% of the mangrove forests have been lost globally at a rate of 2% each year. However, in the recent published paper showed that the mangrove deforestation rate have been decreased in the last decade. Now, this figure shows the drivers of mangrove deforestation in the Southeast Asian region. The main driver were aquaculture, rice cultivation, oil palm plantations, urbanization, and mangrove plantation was not the deforestation, but I think this is the restoration area, which are in pink. And others were like natural disasters and diseases. Now, also it is very clear from this figure that the different countries shows different main drivers for mangrove deforestation, such as in Myanmar, rice cultivation was the main driver. But if we see the Kalimantan and Sulawesi and then Philippines, main driver was aquaculture farm. And if we see Malaysia region, oil palm plantation and urban development, especially like a port development, was the main cause for mangrove forest deforestation. Now, mangrove deforestation and degradation lead to carbon emission, and which was stored in the soil for a long time. However, it's not only we lost the carbon emission happens, but we also lost the carbon sequestration potential due to deforestation and degradation. And therefore, global warming can accelerate at twofold rate. Though the mangrove forest covers only 0.7% land cover globally, but it contributed 10% of the emission from deforestation globally. Now I will show you some results from my studies. Like my first point is a nationwide mangrove forest carbon inventory across the different land use types and environmental settings needed to understand their role in climate change mitigation. Because if country really wants to include mangrove forest in nationally determined contributions, carbon credit system, and ecosystem services, it is better to understand mangroves at landscape level due to their dynamic in nature. And also all mangroves are not same due to different species composition and in geomorphological settings. Also, different land use types are important, such as deforestation, degradation, and restoration, where mangroves need to be sampled. Now, this this research is about the impact of degradation, deforestation, and restoration on mangrove ecosystem carbon stocks across Cambodia. Now, to quantify a carbon stock, a sound, internationally recognized and accepted methodology is needed, because still most of the countries are using the default values from IPCC wetlands action. And for better forest reference emission level calculation, it is good to collect better data using a standard methodology. So we used the SWAM protocol developed by C4, and then we measured all the 5 main components for the carbon stock quantification across Cambodia. Now, we used a random gridded sampling methodology across Cambodia. So in total, we sampled 46 plots, and one plot is comprised of 5 subplots. So in total, around 230 subplots of 7-meter radius circular plots established. And we can see from this figure that the mangrove carbon stock decreased from northern to southern significantly. And if we would have sampled only one area or one local area, we could easily over or underestimate the carbon stock of mangroves in Cambodia. And therefore, a nationwide carbon inventory is needed. We can see from here our mean carbon stock for Cambodia was 788 megagram carbon per hectare. Now, because northern region was in Kokong area, and in Kokong mangrove area, 23,000 hectares of mangrove are under protection, and that's why they show higher mangrove carbon stock. However, if we move from central to southern, the carbon stock decreased. It's because due to development, tourism, or illegal selective logging. And also one of the region was southern region mangroves are not getting enough rainwater or fresh water input. And because local people are using rainwater for agriculture purposes, and that can easily increase the salinity for the mangrove, and that can lead to slower mangrove growth. Now, it is clear from the figure that the deforestation reduced the carbon stock by 60%, though there was no significant difference between intact and degraded carbon stock. But it is clear that the degradation do lead to some amount of carbon emission. We also sampled 25-year-old restored mangrove forest. And it is clear from here that 25-year-old restored forest carbon stock is similar to intact forest in terms of carbon, and which can offset carbon from deforestation and degradation. However, conservation of remaining mangrove should be the effective strategy for climate change mitigation and adaptation. Now, it is clear from our previous slide that if the effective restoration was done, it can provide similar ecosystem services in between 25 to 30 years, depends on the species planted or environmental setting or different land use types. Therefore, the purpose of mangrove restoration should be clear, and effort should be focused on formerly deforested or degraded areas rather than other ecologically important ecosystems. Now, this study was done in Philippines, and for growth performance and structure of a mangrove afforestation project on a former seagrass bed in Mindanao, Philippines. Now, it is clear from this figure that the mangrove can grow on seagrass bed and shows the same above ground biomass as compared to other studies. Those mangroves were planted in different deforested or degraded areas. Though mangrove planting in these areas could eventually lead to seagrass loss. This is clear from this figure. And loss, which is due to the canopy area cover. If higher canopy area cover, the seagrass need to compete for sunlight, and eventually it will die. And because each ecosystem provides a specific ecosystem services, which is very important for the whole food chain that includes plant, animals, and microbes. And this whole chain can be easily disappeared due to the wrong plantation. Now, most of these restoration efforts have been through outplanting of Rhizophora species seedlings, many of which have failed because the proper hydrological and ecological conditions were not properly assessed. And also, after the restoration, no proper monitoring was done. So therefore, a long-term ecological monitoring program is needed to understand the combined impact of anthropogenic and global climate change, such as sea level rise and typhoons, on mangrove forests to better conserve and manage nature-based solutions. So this paper shows the litter fall dynamics in mangrove stand over 5 years in Okinawa. Now, in this study, monthly litter fall was collected from April 2005 to March 2010. And this figure shows the clear pattern of total litter fall production. So in summer, it was highest, and in winter, it was lowest. Leaf litter fall was the main component and constituted 50 to 70% of the total litter fall. Now we can see some peaks here, very high peaks. and during the summer season, and that was caused by the higher production of branch litter form during the typhoon months. Now, it is clear from this figure that the branch litter fall did not show any clear pattern, but peaked in the summer. And because typhoon occurred in July and September 2006, and also in July 2007, and that have increased the amount of branch litter fall. So it is clear from this figure that the monthly maximum wind speed shows the strongest effect on branch litter fall. So if we would have not monitored the long-term litter fall production, it would be difficult to see this pattern. So if we would have done this study only from April 2005 to March 2006, so only for 1 year, which is fine to have seasonal data, but to capture the episodic event, it is necessary to monitor long term. Finally, I have 3 take-home messages from this presentation. Firstly, the mangrove forests play an important role in climate change mitigation through carbon sequestration potential, and also helps in adaptation to sea level rise through wave attenuation and vertical elevation, and therefore can be act as an essential component of nature-based solution. Protecting and conserving remaining mangrove forests should be priority, and enhancing ecosystem services via restoration and rehabilitation on earlier deforested and degraded areas. And last one, long-term mangrove monitoring studies are needed to understand the combined impact of anthropogenic and global climate change to better conserve and manage these important nature-based solutions. I would like to thank and acknowledge funding agencies, USAID, GST and JICA, GSPS, and NEXT during this research, and also the institutions, University of Hawaii Manoa, Tokyo Institute of Technology, University of Syracuse, and University of Malaya, and United States Forest Service for giving me opportunity to conduct the research. I would also like to thank mentors, colleagues, and students who helped me to collect data and discuss the results. Lastly, once again, I would like to thank the Ministry of Agriculture, Forestry and Fisheries and JICA for giving me this species award. Terima kasih.
シャルマ様、ありがとうございました。続きまして、ダイレクトフィルター PCR を用いた牛伝染性リンパ種ウイルス感染の新規診断検査手法の確立と題しまして、原が丸アリアリエルダウス様にご講演いただきます。ダウス様、よろしくお願いいたします。
Good morning, everyone. Today, I'd like to start my presentation for the today's ceremony by the title, Establishment of a Novel Direct Filter PCR Test for BLV Infection. My name is Hala Gamal Ali Daouz, related to the Graduate School of Medicine and Veterinary Medicine, University of Miyazaki, Japan, and also I'm an assistant lecturer at the Faculty of Veterinary Medicine, Banha University, Egypt. This will be the contents for today's presentation. It will include the BLV background, direct filter PCR, outcomes, application, and finally conclusion. Let's start by the background. Bovine leukemia virus or BLV background. The BLV causes severe chronic neoplastic disease to the infected cows, which called the enzootic bovine leukosis or EBL. As you can see in this photo, shows some of the clinical signs of the EBL disease. So the superficial lymph nodes are obviously enlarged and can be palpable under the skin, which is at necropsy will be infiltrated with neoplastic lymphocytes. The infection is often subclinical and may result in lymphocytosis or tumor. The animals can be infected at any age. However, the EBL are seen typically over 3 years old, elder cows. The number of BLV infected cows increasing worldwide. Here, I will show you just 2 examples, in Japan and Egypt. Firstly, in Japan. In Japan, the nationwide survey of BLV infection from 2009 to 2011 showed that the prevalence of BLV infection in animal level has been continuously spreading year by year. In Egypt, it's starting 2002 and 2018, the serological screening revealed a seroprevalence of antibodies directed against BLV of 37.7% and 20.8% in cattle under 2 years old, respectively. Until now, there is no treatment or vaccination for this disease. The BLV causes severe economic damage to the infected farms due to loss of production potential and culling or death due to cancer. The diagnosis of BLV infection is the first step in identifying and eliminating the infected animal herds and the eradication of EBL. There are currently several methods for BLV diagnosis as BLV ELISA, real-time PCR, and nested PCR. The currently available diagnostic tests, as previously mentioned, are useful for BLV diagnosis but have several limitations in the accuracy and rapidness of the BLV diagnosis. Here I will show you some of the limitations of the BLV already established diagnostic test. Firstly, most of them are time-consuming, which requires several steps, and also cost-consuming. And most of them required blood sampling. So the veterinarians must go to the farms and do the blood sampling by themselves using specific blood collecting vessels like needles and vacuum tubes. And all of them require DNA extraction, which is several steps, which make it like time and cost consuming. All of these limitations directed us to the aim of this study. The aim is establishment of a direct filter PCR as a BLV diagnostic method that simplified the collection and transportation of blood samples and pre-treatment for diagnostic tests. Let's move to the direct filter PCR. Here I will show you the technique of a direct filter PCR. So this photo shows the equipment which used for the direct filter PCR. It's easy and simple equipment. Like the first one is the Unicore cutting pen and commercially available filter paper, and this green bed used for supporting the filter paper during cutting technique. Usually our lab receives the samples like this, dried blood spotted filter paper which used as a sample and template for the direct filter PCR. This photo shows how the cutting pen cut the dried blood spotted filter paper, and only 1 millimeter cut piece used as a template for the PCR. So from these tips, the direct filter PCR is easy, rapid, and simple method for BLV diagnosis. Here I will show you the difference between the conventional PCR, which is already established method for BLV diagnosis, and the direct filter PCR, which I established by myself. Firstly, the conventional PCR required the blood sampling by the veterinarians using specific blood tubes and needles, which is time and cost consuming, and then DNA extraction for the blood samples, which is time and cost consuming. Only one sample requires 2 hours work for DNA extraction, and finally the BLV amplification. However, the direct filter PCR is a rapid and simple method which enables us to collect the blood without blood collecting vessels, just small injury to get the small blood drops on the filter paper, let it dry and used as a template for the PCR without DNA extraction. So the direct filter PCR omit the blood sampling by using the blood collecting vessels, and also omit the DNA extraction, so it's rapid and sensitive method. Let's move to the outcomes. Direct filter PCR is sensitive and specific BLV diagnostic test comparing to the BLV ELISA result. The reliability of the direct filter PCR was evaluated with the real-time PCR, indicating a constantly high degree of the agreement between the two previously mentioned tests. The dried blood samples spotted onto the filter papers were stable for at least 10 days at room temperature. So this method also simplifies the storage of the blood samples, So just the dried blood spot on filter paper can be sent using the mail post at room temperature. So there is no need for 4-degree or refrigerator or minus 20 deep refrigerating for the transportation and storage of these samples. So it's rapid and easy method. This filter PCR method enables us to omit the cost and time of blood collection and also facilitate the transportation and storage process during the transportation process at room temperature, and also without DNA extraction, so it's also omit the time and cost of these steps. Also for the application, the direct filter PCR allows the farmers to collect the blood samples on filter paper and transport them in the mail post at room temperature. This will reduce the risk of disease transmission between the farms. It's not necessary for the veterinarians to visit the farm and use the blood collecting vessels, so the farmers can do the blood sampling by themselves because it's easy method, just small injury and touch by the filter paper, and then let it like 5 minutes or 10 minutes for dryness, and then send for the mail post at room temperature. Direct filter PCR can provide a feasible BLV control strategy and mitigate the economic losses to farmers and veterinarians of developing countries. Additionally, it requires neither high precision instruments nor well-trained personnel to operate, so it's suitable for use in practice and in less equipped laboratories, especially for developing countries like Egypt. In conclusion, I have successfully established a novel diagnostic test for diagnosis of BLV using direct filter PCR method. This is the first report at which the blood-spotted filter paper can be used directly without DNA extraction for PCR reaction for simple and rapid BLV diagnosis. Finally, my research findings of the BLV study have been objectively evaluated by being published in Transboundary and Emerging Diseases journal, which is high impacted journal, like the impact factor is 5.005, which is a peer-reviewed journal that considers articles on all aspects of infectious diseases of greatest economic threat to animals and humans. Acknowledgement. Firstly, I would like to thank Allah for giving me the chance of life and all chances of success during my studies. Then, I would like to express my sincere gratitude to my advisor, Associate Professor Dr. Satoshi Sekiguchi, for his continuous support of my PhD study and research, for his patience and motivation. Besides my advisor, my sincere thanks to Professor Dr. Johnson Romini for his continuous encouragement and insightful comments. Also, I'm grateful for the support and recommendation of Professor Dr. Muhammad Ghanem, the Dean of Faculty of Veterinary Medicine, Banha University, Egypt. This is my faculty in Egypt. Also, I thank all my fellow lab mates in Animal Infectious Diseases and Prevention Laboratory, or AIDP Lab, for their help, support, and for all the fun we have had together. Last but not the least, I would like to thank my family members who supporting me spiritually throughout my life. Thank you for listening.
ダウス様、ありがとうございました。では最後に、グリニシディアとの観察システムにおけるカカオの窒素栄養と題しまして、ジェームス・セウトラ・カバ様にご講演いただきます。カバ様、よろしくお願いいたします。
My name is James Sitra Kaba. I'm a lecturer at the Kwame Nkrumah University of Science and Technology in Ghana. My presentation is on nitrogen nutrition of cocoa and intercropping systems with Glacidia. Ghana is located in West Africa. So what you see here is a map of Africa, and Ghana is located here. And if you take the map of Ghana, all these green parts are regions where cocoa is produced. This is a cocoa tree, and these are the pores from cocoa plants. Now, Ghana is the second largest producer and exporter of cocoa after Ivory Coast. 1.6 million acres of Ghana's land is under cocoa cultivation, and about 25 to 30% of Ghana's population depend on the cocoa sector alone as their source of livelihood. So you usually hear statements like Ghana is cocoa and cocoa is Ghana, just to emphasize the importance of cocoa to the livelihood of the people of Ghana. Apart from employment, which it offers about 6 million to Ghanaians, then it's also a major source of foreign exchange. Ghana currently earns about 2.5 to 3 billion annually from cocoa export. Chocolate is produced using cocoa as a raw material, and chocolate is widely consumed throughout the world. Other products are also produced from cocoa, so the importance of cocoa cannot be overemphasized. Current production, Ghana's production is about 450 kilograms per hectare compared to other countries who get about 800 to 1,000 kilograms. So one will ask himself, or the question that arises is, why such a low yield for a country that is considered the second largest producer and exporter of cocoa? Two things. One has been the impact of climate change and also low soil fertility, especially nitrogen, because of continuous cropping. Nitrogen deficiency usually causes the cocoa plant to be stunted in growth. The pores are poorly formed, the fruit get aborted before they mature, and all these factors contribute to the low yield that farmers get at the end. There has been interventions. In 2012, the government introduced nitrable fertilizer, which is a calcium nitrate-based fertilizer. And the idea was to improve upon the soil fertility so the production level can increase. Since 2012, effort has been made through the Cocoa Research Institute of Ghana to approve and launch several fertilizers, and the media has appropriately reported that. However, I must say that the impact of these introduced inorganic fertilizers have not been felt by the farmers because yields are still lower. And one reason is that in Ghana here, most of the cocoa farmers are smallholder farmers, about 80% of them. And this inorganic fertilizer is usually too expensive beyond their reach. So you see them calling for government to subsidize it so the price will lower. Another problem has to do with poor road access to cocoa farms. Where these cocoa farmers stay is far from where their farms are located. So how to transport this inorganic fertilizer to their farms is difficult because the roads are not good. What you see here is a road leading to one of my study sites. Another problem has to do with the unsustainable nature of the subsidy that they call the government to subsidize. Farmers will have to train how to appropriately apply these fertilizers. And lastly, even when government subsidizes these fertilizers, it's being smuggled outside the country and sold at a higher price. So the cumulative effect is that only 20% of our land area collected under cocoa cultivation receive inorganic fertilizer. And the current report also indicates that about 17% of farmers in Ghana apply any form of inorganic fertilizer. What is my alternative solution? My alternative solution was to identify a tree, a Glacidia sepium, which is usually called the mother of cocoa, because most farmers use it to intercrop with cocoa. So what you see here is Glacidia tree with intercrop with cocoa. And my research concept is this. The atmosphere we know contains about 80% nitrogen gas. However, this nitrogen cannot be used by plant, okay? But legume plants like Glacidia have the potential to form symbiosis with rhizobia so that they can fix the nitrogen into a form that the plant can utilize. So my concept is that if the legume plant Glacidia is intercrop with cocoa, Glacidia can form symbiosis with rhizobia. It can fix the nitrogen. The nitrogen remains in the shoots of the Glacidia tree. When farmers prune these shoots and apply it as organic manure or green manure, it will decompose, release the nitrogen, and the other nutrients in the cocoa plant can take it up. So my first idea was to estimate the amount of nitrogen that Glacidia tree can fix. So I used the 15N natural abundance technique, which is the most appropriate method to use for tree crop estimation under field condition. And the technique simply compares the delta 15N of a plant that fixes nitrogen like Glacidia to the delta 15N of a plant that does not fix nitrogen like cocoa, which we usually refer to as the reference plant. So what are the assumptions of the 15N natural abundance technique? The assumptions are that if a tree gets all its nitrogen from fixation like a legume tree, then the delta 15 would be almost like 0. Now, the second assumption is that if a tree gets all its nitrogen from the soil, then like a cocoa tree, then the delta 15 value will be between 5 to 8. And the third and the last assumption is that if a tree gets part of its nitrogen from the soil and part of the nitrogen from fixation, like having a glaciated tree under full condition, then the delta 15 is likely to be between 0 and 8. So with the help of this formula, one is able to estimate the presented nitrogen derived from the atmosphere. So what you see here is a summary of the analysis. I took samples of cocoa and glacidia, and with the help of an isometric mass spectrometer, I was able to analyze for the delta values. I actually worked on 5 study sites in 2 regions, eastern region and the western region of Ghana. And the data was taken in the dry season as well as in the rainy season. Apart from site 3, all the other sites show significant difference, and I was able to estimate the presented nitrogen derived from the atmosphere. So my estimation shows that the nitrogen derived from the atmosphere by a glaciated tree ranges from 27 to 60% of total nitrogen, based on the estimation that I made. Now, it is not enough to know the presented nitrogen derived from the atmosphere or the concentration of nitrogen in the shoot biomass. You need to know how much new above ground biomass that a farmer can get from the glaciated tree after pruning. That will translate to the amount of nitrogen that the farmer is likely to incorporate into his soils. So to establish that fact, I adopted a method to estimate how much new above ground shoots a glaciated tree is able to produce in a year. So what I did was to tack a glaciated tree after I've pruned the whole shoot, attack the beginning of a new growth. Then what you see here is that after 12 months, I was able to estimate how much new shoot has been produced. So what you see me here holding is the new above ground shoot of a glaciated tree after 1 year. So this is a summary of the results. So the total shoot was 37.22 kilograms per tree. The amount of nitrogen in that total shoot was 1.56 kilograms per tree. And remember, I did say that my estimation shows that 27 to 60% of the nitrogen in the total shoot. So this nitrogen is from N fixation. So that translates to the amount fixed to be 0.4 to 0.8 kilograms per tree per year for glaciated tree. So my next question was to find out which rhizobia was responsible for the N fixation that I've just established. So what I did was to dig the root, the soil under the glaciated tree. In the dry season, I didn't find nodules. However, in the rainy season, I was able to establish the presence of nodules. So what you see here are root nodules of glaciated tree. So these are the root nodules. So this is how the nodules of the glaciated tree looks like. So with these nodules, I processed them, and with a cultured independent method, I was able to molecularly characterize them and identify that Rhizobia tropici and Rhizobium ele were the 2 major Rhizobia species in the root nodules of Glacidia and responsible for the fixation. My next target was to see how farmers can utilize the large quantity of biomass that usually exists or present in the floor of cocoa plantations. So what you see here is a cocoa farm with a lot of litter under it. And I must say here is that like cocoa litter are usually high in carbon, lignins, polyphenols. That makes it very difficult to decompose, making decomposition very slow. So my idea was, how can a farmer who practices an intercropping system harness this litter as source of organic manure to improve upon the soil fertility? So what I did was to conduct a simple decomposition and mineralization experiment using litter bag technique. So I trapped litter under cocoa, then I pruned the glaciated shoots. I designed this litter bag, and at this point, what I did was, after designing the litter bag, I had 3 types of litter. One was sole cocoa litter to mimic what happens under cocoa monocropping system, and a mixed litter, which was 40% glaciated shoots, 60% cocoa litter, to mimic what happens under intercropping system. And the third one was whole glacidia shoots. So the litter bags were buried under several cocoa trees like this. Then the decomposition rate was estimated. Now, what you see here is the decomposition pattern of the biomass. So you have the mixed litter decomposing at a faster rate compared to the cocoa litter. Now, if you look at the litter types at 126 days, all the glaciated shoots had virtually decomposed, whilst about 70% of the mixed litter had decomposed, reflecting what happens under intercropping system. The cocoa litter had little decomposition. The structure was still intact, also mimicking what happens under cocoa monocropping system. The pattern of nitrogen release clearly shows that after 50 days of bearing the litter bags, significant amount of the nitrogen for the mixed litter had been released, while the cocoa litter took a longer period to release its nitrogen. Now, my question was, how can this experiment be linked to farmers' existing knowledge to enhance adoption? So I got funding from GCRF in collaboration with the University of Quebec. I had a colleague there, and the idea was to translate this innovation or this experiment into a compost production system where farmers can produce their own compost and apply it as organic manure. So my first idea was to go to the farmers and find out from their understanding of shade trees and the role shade trees can play in the adoption of cocoa agroforest system. So I visited farmers, had meetings with them, questions were asked concerning the importance of shade trees, what they understood by the importance of the shade trees and its contribution to their cocoa farms and the cocoa yield, and also suggestions from them as to how to improve cocoa shade trees incorporation into cocoa farms. I must state here that the results clearly indicated two things. Farmers had a very good understanding of the direct benefit of shade trees, like the role it plays in terms of medicine and the food that they get from it. However, the indirect benefit like nutrient recycling and climate change mitigation or adaptation benefit, farmers did not have knowledge on that. So the conclusion was that there was a need for a stakeholder collaboration between farmers and stakeholders in the cocoa industry, so that farmers can be educated as part of cocoa extension education to appreciate the indirect benefit that they could get when they incorporate shade trees onto their farms. Now, my general conclusion implication, my research have implications for soil nutrient recycling, agroforestry waste management, and sustainable cocoa production. Finally, co-benefits such as mitigation, adaptation, and adaptation against climate change caused by the removal of shade trees and burning of agroforestry waste can also be achieved. Remember, I did mention at the beginning of my presentation that there were two factors that were resulting in low yields in cocoa farms. One was climate change impact, and another was low soil nitrogen or low soil fertility. And I believe that if farmers are able to integrate the right shade tree, they will be able to get higher yields because the soil nitrogen content will be improved. And also other benefits like shade trees serving as climate change mitigation adaptation strategy will also be achieved. I want to use this opportunity to say a very big thank you to JICA and also the Ministry of Agriculture, Forestry and Fisheries of Japan for the honor done me. I'm so grateful for the award. As an early career researcher, this is a big boost to me. Thank you so much. I'm very grateful. Bye-bye from Ghana.
川様、ありがとうございました。以上をもちまして、2020-2021年若手外国人農林水産研究者表彰ジャパンアワードを終了させていただきます。ご参加いただきまして誠にありがとうございました。